Good morning. Welcome to this Southwest Marine Ecosystems webinar. And today is fish and turtles, so I'll get straight in. Been a very frustrating year, not being able to get down to the shore or out on the sea or under the sea for many of you, unfortunately not for me. And particularly in my case, not being able to get out to the fish market for most of the time. I had about 3 months when I could get down there and that was it. But so it's, and also, the lack of time of people being out at sea or on the shore does mean we seem to have got a lot less records this year. So I'm trying to patch together what I've got from the records so far. Please do send records of fish and turtles to me so we can make a really good 2020 annual report for Southwest Marine Ecosystems. Well, this young gentleman started off well. Out on, I presume, New Year's Eve, and right at the beginning of the year, caught this fish. It is a blue skate. The skates were separated about 8-10 years ago into two species, blue and flapper. This is the smaller of the two, tends to be the more southern one, but still, as with all the so-called common skates, the blue Dipterus batis and the flapper Dipterus intermedia, both are critically endangered. And this fish photographed and put back and swam away quite happily. But a lovely start to the year. Critically endangered species found to be living inshore off Chesil Beach. An endangered species that is very much associated with the Southwest is the basking shark. April was an astounding month for the basking sharks. 19 sightings around Cornwall and one in Plymouth Sound. But unfortunately, the rest of the year didn't follow that pattern. I've only been so far got 7 reports of basking sharks from the whole of the summer. And the final one for the year was unfortunately a juvenile washed up on the lizard in late November, tangled in fishing gear. So brilliant start, but poor finish to the year for the basking sharks. Thresher sharks, been getting quite a few of these around Torbay lately. This one was in June, early part of the season, and a very young one. It was only about 2 meters, which is pretty small for a thresher. So this was seen in Torbay. One was seen in July off Jenny's Cove in Lundy. By August, the numbers around the Isle of Wight, which seems to be a centre of abundance, had built up, and one estimated at 100 kilos was caught. The very same day off Plymouth, another large one was hooked, but it got away. So we don't know how big that was. In fact, in October, a thresher was caught over St. Catherine's Deep, just south of the Isle of Wight, which was thought to be estimated at 280 kilos. The UK rod and line record is 150 kilos. This one won't count because to count for records, they have to be brought ashore and weighed ashore. And that, of course, means killing the animals. And the trend, fortunately, amongst anglers these days, they often seem to think they've caught fish bigger than the record, but they suspect the lives of the animals and release them rather than claim the animal. They just get the satisfaction for themselves. It's great to see this trend coming up in anglers these days. You know, people like to catch them, see them, but also love to see them go back alive to swim on for the future. Another one that went back alive, caught by a trawler in Start Bay, was this sturgeon, a young one, only 1 meter long. The sturgeon, European sturgeon, is a critically endangered species. There's only one known breeding population in the world, which is in the Gironde, in France, and it's all the ones we have are coming from that one site. So a really critically endangered species. Lovely to see that young ones are reaching our shores, and the fishermen these days the commercial fishermen are aware and try to get them back alive wherever possible, as is the case here. In fact, over the last few years, there's been a number of sightings in this area: Start Bay, Berry Head, Start Point, Lime Bay. It looks as if we may have an marine aggregation area, possibly a very good feeding area for this critically endangered species just off the shores of South Devon. So we've got some really wonderful rare fish here. Well, we've always known we've had seahorses around, and it was an absolutely amazing year for seahorses. The first was one that was filmed in Poole Harbour. Any photography and filming of seahorses has to be done under a license. I think it's from the MMO, either MMO or Natural England. But anyway, they're done under license, and Steve Truella managed to film a short-snouted seahorse on the 5th of May. On the 6th of May, a spiny seahorse, Hippocampus gutulatus, was found at Sandbanks, also in Poole Harbour. Then throughout May, spiny seahorses, Hippocampus gutulatus, had their best year yet recorded in Studland Bay, which is, of course, a newly designated marine conservation zone. A total of 46 individuals were recorded. Highest in a year previously was 41. 21 on a single dive. So the population and stocks there were looking very good. Unfortunately, when the first lockdown ended, there was a massive influx of boats and people to Studland Bay, increased disturbance, anchoring amongst the seagrass, and the number of seahorses found immediately after that went down to 3, from 46 to 3. Also in May, 16 spiny seahorses were seen and filmed under license again at an unnamed site in Devon. So there are other sites apart from Studland, Poole Harbour area, Poole Bay, and some sites in Devon seem to be good. An unidentified seahorse was seen at Sidmouth, also in late May. The last of the season was a spiny, seahorse at Studland on the 12th of October. So 5th of May, starting with a short-snouted, through to a spiny on the 12th of October. Quite a long season. This Southend dory was caught south of Plymouth. Sorry, I can't remember exactly when. I think it was about August time, and was probably the 12th recorded in Britain. The first 2 records are just strange-looking dory landed at Newlyn. So 10th since it was recognised as a British species, probably 12th overall. All the records, it's a species that's been shown to be moving up from Spain, Lusitanian area, through Biscay to Ireland and Britain over the last 25 years. And I say now we've had 12 in British waters, and the last 2, this one and one the previous year, have both been from the Plymouth area. All the previous ones were western approaches, Isles of Scilly, or the Cornish coast. So one of the easternmost of these, there's another species, the rosy dory, has not yet been found in British waters, has moved up the Atlantic coast of Europe in the same way, and now 1 or 2 have been found off the Irish coast. So hopefully that may turn up in Britain before too long. The comber is a ceranid, a member of the grouper family. Now, some of these can go to 3/4 of a ton or more, but the comber is just a small one. That's quite a reasonable sized one. It is one of the northernmost ones in the East Atlantic. And normally we get 1 to 3 turning up over about a 4-year period. However, this year there have been 4, sorry, not last year, 2020, there were 4. Ben Lowe, who fishes off the north coast of Cornwall, had 1 in May, followed by 1 in June. There was 1 off Guernsey in August and 1 south of Prawl Point in September. A more southerly species, hopefully becoming more common, population establishing itself. So good to see those turning up. Ray's bream. This is an oceanic fish that turns up in our waters from time to time. When it turns up, the most usual pattern is vast numbers off the Irish coast, sweeping around the north of Scotland and down into the North Sea, where they'll turn up for several years until the population peters out. So we don't get many in the southwest. Often when we do, they tend to be forerunners or just outliers of one of these mass immigrations that's happening. However, we had 1 in 2019, 1 2020, but no sign of large numbers building up as yet, but it could be something that's coming. The variable blenny, a lot of the books call it a ring-necked blenny, and I can see absolutely no reason to do that. They are quite variable in appearance, but nearly always recognizable by this honeycomb pattern on the cheeks and the fine, much branched tentacles over the eyes. Pilicornis, all these fine tentacles over the eyes, but also this honeycomb pattern on the cheeks is quite distinctive. First discovered near Plymouth photographed near Plymouth in 2007. It's now well established and breeding in the Plymouth area. Also looks though there's a group around Babbacombe in the Torbay. However, now getting records from Cornwall. And in 2020, there were continuing to 2 records from Dorset, following on the pattern there. And in the Channel Islands, 1 from Sark, having previously been found in Guernsey. So building up in Sark as well. This is one of the easternmost ones known, one on a wreck in Eastern Lyme Bay. You can see why it's called variable. You can just about make out that honeycomb pattern on the cheeks. This is probably male coloration in the breeding season. So lovely picture there from Mike Markey of this one on a wreck in Lyme Bay, and showing we get males in breeding coloration, we presume, here. I hope you can see this fish. It is tiny, pushing up against that sponge there. It is Gillette's goby. Very few records of this species. It's probably the smallest marine fish on the Atlantic coast of Europe. Very few records, very little known about it. But is that because it's very rare, or just it is so small at 2 to 3 centimeters that it's overlooked? Nick Owen has found several of these. He's brilliant at it. So very useful to have somebody who's got such good eyesight diving regularly and submitting his records to Sea Search and to myself around our coasts. Well, from small to big. At one time, bluefin tuna was the largest fish ever caught on rod and line in British and Irish waters. The 6-gill shark has taken over that record now, but bluefin tuna now turning up around our coasts. Obviously, Tom will be talking about these later. This photo from Guernsey, but good numbers around. The earliest date I've got recorded is the 14th of June. And Tom's not going to beat my latest date recorded for the year, because that was the 31st of December. Sailfish. 14th and 15th of August, there were 2 reports from anglers and from bird watchers around the Porthgwar area of southwest Cornwall of sightings of sailfish. There was also, really close, within a few meters of the rocks, a further report on the 29th of September. However, there's only ever been one confirmed record of a sailfish in British or Irish waters. So without good photos, at least, or better still, a specimen, it's probably these were misidentifications, the most likely thing being that they were swordfish rather than sailfish. Swordfish, more stockier, broader bill, and without just a small, more dolphin-like fin, not that great big sail fin. Any records of these are so useful, because if they really were sailfish, then building up these early records is so useful to see the pattern if they do become more common. The largest bony fish in the world, we've gone from one of the smallest to one of the largest, the sunfish. These seem to have been fairly common in 2020, mainly July and August. There were 16 recorded around the Isles of Scilly in that period. So it seems to have been a reasonable year for them. I haven't got that many records in yet, but hopefully when more come in, be able to build up a more detailed picture. One most interesting thing, on the 27th of July, in the Scillies off Deep Point, St Mary's, Martin Goody was able to film a grey seal predating on a sunfish. So an interesting record there. A little outside our area, but just across the Bristol Channel at Porthcawl, a zebra moray. It's an Indo-Pacific species, so obviously not a natural occurrence. This fish, say zebra moray, Gymnomurena zebra, it must either have been deliberately released, and there have been 1 or 2 fish this has happened with, escaped from a private collection, or dumped after it died. I once, some time ago, had a South American armoured catfish trawled up off the south coast of Cornwall. It was dead at the time and presumably had been flushed down a loo after it died and washed out to sea. Whether such a thing might have happened with this or just simply dumped, but it's something we need to look out for. And give some indication of the size there and the choice of item to illustrate the size, full-size bottle. I had a call from a fisherman friend while he was out at sea that one of his friends had caught a strange fish. They hadn't a clue what it was. So it was sent into Plymouth Fish Market for me to go and look at. Came from the Swift Shore. It was caught off Wembury in Devon. And I recognised immediately that it was a cyanid, a group called the croakers and grunts, mainly tropical, subtropical, and particularly found around North and South America and the Pacific coast of the Americas. I had been given
9 years before, this photo taken at Plymouth Fish Market, but I never received the fish. I wasn't unable to find out where it had been caught. And I sent the photo to the world expert on cyanids, and he couldn't identify it for certain. So when I was able to compare it with the present specimen, I was able to get an identification of Micropagonius undulatus, the Atlantic croaker, a reasonably common fish in the northwest Atlantic, particularly around the Gulf of Mexico and the southern states of the USA. So this is the first definite record. The previous one appears to be a record of it as well, but the first definite record of it from British and Irish waters. Checking around with colleagues, it has never been recorded in France or Spain or Ireland. So it's hard to see it coming across the Atlantic to the warmer waters and moving up. However, there have been occasional records over the last 6-8 years of them occurring in estuaries of Belgium and the Netherlands, where they're thought to have arrived in ballast water. So there may be a population building up over on that side of the North Sea, and this fish may have come from that area. Be great. It's in my freezer at the moment, waiting after lockdown to go to the British Museum, and hopefully the testing carried out on DNA or stable isotopes can tell us where it came from, whether it's from one of the low country, the North Sea population that's building up now, or from the Americas. So a big question there, where's that one come from? Moving on briefly to turtles. I contacted Rod Penrose, who keeps the British and Irish turtle records, and in the whole of 2020 they had received 8 records for the whole of Britain. I'll come back to that in a minute. From that, I got 2 records. The hard-shelled turtle that was released with a poor quality photo, so it could be leatherback or Kemp's, most likely, sorry, not leatherback, loggerhead or Kemp's, possibly green or olive ridleys, but most likely loggerhead, was tangled in fishing gear at Budleigh Salterton, successfully released and swam away alive. And but a badly decomposed leatherback was found on Briar Island in the Scillies in October. I've since had from contacts in the Scillies 2 further sightings of live ones around St Mary's and St Martin's. So we now have 4 records for the Southwest. If anyone knows of any others in the Bristol Channel, the English Channel, down to the Channel Islands, please let me know to go into the report. As I say, 4 this year for the Southwest. Only 2 of those were actually in the national record base, so that's been built up to 10 now. But compared with previous years, in previous years, the marine turtle report of strandings, well, in 2017 it was 37. 18 is more usual. 8 is ridiculously low, presumably less people out to find or to sight at sea. So our pattern for the Southwest fits in with that. 13 in 17, 19 or 20 in 18, 14 in 2019, and just the 4 this year in 2020. So hopefully we'll get some more records in to build up a better picture. All of the ones in the national database were leatherbacks, I believe, and that seems to be the dominant one. Well, gathered my information from lots of sources. Can't name quite everyone, but hopefully most people who've helped me out are named there. So, questions, and please do send sightings to me, all turtle sightings and any interesting fish ones. please send douglas.herdson@btinternet.com. Thank you.
Okay, thank you, Doug. I don't know if you want to share your camera.
I'm trying to.
Questions. There we go. Brilliant. Okay, so we've got a few questions already coming up. So first question. So Nigel Mortimer has said, Doug, is there something that could be done to reward those anglers that are more sympathetic to their catch, putting them back alive and not claiming a record, etc.? In my ignorance, maybe a new second record size system that can take over and become the norm, discouraging the original dead fish system.
Sorry, I'm just trying to get my screen enlarged. What have I done wrong? I've now
hang on, do you want to stop showing your screen?
Oh, yes, of course, that would do it. Okay, thank you.
No worries. Okay, did you catch that question then?
Yeah.
Okay.
There is, in lots of fishing clubs today, an ethos of we want to see good fish, and we'll see good fish by maintaining the stocks. No, there's lots of there's one charter boat skipper I know who's going after big sharks, and when he gets them on board, he's irrigating them with hoses, keeping them there, just measures them, never lifts them up to weigh them, because one of the worst things you can do is hanging a fish on a hook to weigh it, because particularly with the elasmobranchs, all the internal organs slump down in the body, ripping the mesenteric blood vessels. You release the fish, it appears to swim away, but it's slowly bleeding to death internally. So most people are now learning, support the fish and you catch them out, keep them damp and back. And this does tend to be with a lot of the regular anglers, the ones in the major clubs and that, is the ethos today. Hopefully that will spread. I quite appreciate what you're saying about needing some other way of claiming records. There isn't yet, but most people do kind of accept these unofficial ones. What's done generally with sharks is to measure the length and, if possible, the girth, and from that calculate what the weight would be. I'm sure Simon can actually answer that question much better than me.
I think Simon's actually putting his hand up now, so he might have an answer to that question. Do you want to, Simon, I don't know if you wanted to...
yeah, just very quickly, the Shark Angling Club of Great Britain now accepts estimates of length of fish by the side of the boat for many of its trophies. We try to encourage not boarding of the boat, the fish at all.
Brilliant. Thanks for that, Simon.
With Telios, you do have the problem, or any fish with a swim bladder, you do have the problem that when you bring them up from depth, the swim bladder expands, often pushes the stomach out of the mouth, and those fish aren't going to recover. If you put them back in the sea, they'll be swimming around belly up, and the gulls will take them. I know Ben Lowe, who caught a couple of the combers, was able to put one back, but the other one was blown, so he couldn't. So bringing up bony fish, particularly Telios, with anything with a swim bladder from a depth, they're not going to survive, unfortunately. But if they are going to survive, then yes, please put it back. And so this is becoming the ethos.
Okay, thanks for that, Doug. So Keith has just put a reminder, it's not a question, but please clarify for participants, it's an offence to deliberately seek and then photograph seahorses without a licence from the MMO. If you come across a seahorse, do not photograph it. Flash guns are advised against, but not prohibited. And he'd appreciate any corrections to his understanding. Charlotte's agreed as well, that's her understanding there. So that was more a sort of point. I think you mentioned that really,
I mentioned it
but
3
I think
times,
just to clarify that. Yeah, to clarify that is an important point. Okay, Paul Summerfield has asked how you distinguish Parablennius ruber, the red blenny, from the varial blenny, or are they the same thing? Are they the same species?
No, they are very different and very different habitats. Red blenny is very much like a tompot, but with a slightly thicker tentacle over the eye, and
bright red colours, tends to be found in slightly deeper than you would a tom pot, and areas where a lot of movement, water movement, more currents. Whereas a variable blenny tends to be in shallowish waters, quieter waters, and particularly those little tentacles over the eyes, little antennae and whatever, over the eyes are very fine, like a series of hairs. I find the thing at glance is that honeycomb pattern on the cheeks, but it is the antennae over the eyes are totally different in pilicornis to what they would be in ruber, the tom pot, or of course Montagues.
Okay, thanks for that, Doug. We've got a few more questions popped up. We're running out of time a little bit, so we have to take these ones quite quickly. I think they're quick ones. So firstly, in normal years, this is Jenny Hickman's asked, in normal years, do most turtle records come from strandings or sightings?
It's usually about 50-50. Now, Cornwall with their Marine Strandings Network pick up a lot of stranded ones, but charter boats out angling and people going dolphin watching, that kind of thing, wildlife safaris, they all pick up sightings. Normally leatherback, but some occasionally of hard shells being more difficult to identify as sightings. They're best identified when they're on shore, but leatherbacks, an upturned rowing boat is the general description.
Okay, brilliant, thanks. Terry Kane has asked, do you receive records from Cornwall Records Office? I've submitted several records of bluefin tuna and sunfish last year via the Orcs app. I'm
in touch with IRCIS, the Cornwall Wildlife Trust's data system, and I haven't yet received them, but they have agreed. They normally do send them through to me, and I've just put in an application for those now.
Brilliant.
In fact, the basking shark data did come from that. Yeah.
Okay. And I should say as well, Cornwall Wildlife Trust are involved in the Southwest Marine Ecosystems Programme. So I'd be very surprised if there wasn't a good link.
Well, they've got two major systems. There's the Marine Strandings Network, who go out and look at what gets washed up on the shore, and there's also Seaquest Southwest, which was a general Southwest thing, but now is just run by Cornwall Wildlife Trust, doing sightings.
Okay.
They've got sighting systems and a stranding system, both run by CWT.
Okay, brilliant. So yeah, hopefully all that will feed into the report. Last question then. So from Susan Fraser, doesn't it hurt the mouths of the fish when they're caught?
That
might not be as simple as
this. Probably yes.
Okay.
I can't see that it doesn't. But what is pain? And I think in us, anticipation of pain is a big part of it, and remembering afterwards. Now, fish have got long memories. The 3-minute goldfish is a complete fallacy, but if they've not got perception that it's going to be painful, it's probably not as bad as we would imagine it.
Okay, that's a good concise answer. I mean, you could probably debate the issue for quite a long time, I think.
I can direct you to about 10 papers, one way and the other.
Yeah. Okay. Well, the question is in the Q&A as answered now, but if any other participants or any of the panelists would like to contribute thoughts or share any papers or any other insights in that, then feel free. Okay, I think we better move on now. So we've got Simon Thomas speaking next about the return of the blue shark. So are you there, Simon? Brilliant. Okay, you ready to? Okay. Feel free to share your camera if you want as well, Simon. So with you.
Okay, you should be seeing me now.
Yep, we can see you.
Yep, you got my slideshow?
Yeah.
Okie dokie. Unfortunately, I've got this at the top of the screen, which makes it more difficult for me to share. 1 second. I need to move everything. This is unfortunate.
All right.
Slideshow, here we go. From beginning. Okie doke.
Great.
Hopefully you should be able to see me now.
Yeah, that's great. Thanks,
Okay.
Simon.
Right. First of all, I'd like to thank Doug for all the work he does. Some of the stuff in this will be about how scientists and anglers don't cooperate, but I mean, the work Doug does is a huge exception. So welcome all to this talk on the return of blue sharks to UK waters. And though my name is on this talk, none of this work would actually be possible without the help of the Shark Angling Club of Great Britain, the Sportfishing Club of the British Isles, and the many skippers who have trusted me with the data, and my fellow scientists such as Paul Summerfield and Tim Smythe, BML. I would very much like to thank John McMasters for the huge amount of effort he does behind the scenes putting this stuff together. The work is unfunded, so very much a labour of love. 10 minutes doesn't give me a lot of time, so I'll dive straight into the presentation if that's okay. Who am I? Well, I was very lucky to spend most of my childhood by water, whether fresh or salt. I spent many of my summers in this lovely little North Devon house between right on the cliffs between Hartland Key and Point, where I was either swimming, climbing the rocks, rock pooling, or fishing. I'm an angler, I'm a crew member, I'm a skipper, and I've seen or caught and released over 4,200 blues myself. I've seen blue sharks at its very lowest point, and then I've seen the return of these amazing creatures to our waters, sometimes in absolutely astonishing numbers. My qualifications are really irrelevant, but they're there for you to see. I've been involved in shark tagging programs both in the UK and abroad, and I am the honorary biologist for both the Shark Angling Club of Great Britain and the Sports Angling Club of the British Isles. I use this platform to promote safe handling and best practice for sharks, which Doug touched on, to ensure the survival of captured fish. Right, why collect data? Very good question. I would say the skippers I collaborate with spend thousands of hours at sea each year after these fish, and the knowledge they hold makes them experts by experience. These people are the real experts in these fish, and their knowledge base is vast. Whether you agree with angling for sharks or not, undoubtedly the biggest threat to sharks worldwide is commercial overfishing, and the latest research has shown a 70% decline in some stocks in the last 50 years. Whilst many countries have an active angling scientific collaboration to enable stock decisions on sharks, the UK does not. And despite the great work of some charities such as the Shark Trust, ICAT, who controlled the tax on many migratory sharks, are often slow to act and often cite the lack of or contradictory data from the unmotivated commercial fleets. As we're a catch and release fishery, our data has a low impact. And as mentioned before, we use our project to inform and adapt safe handling guidelines. And we work with bodies such as the Shark Trust. We have, or we should have, highly motivated participants and long-term, highly accurate data. The Shark Angling Club of Great Britain has data from 1953 to present, and the database now is over 100,000 blue shark captures. And there's an effort component, crucially, in the number of trips that these sharks were caught on. They have also participated in several tagging studies and contributed to many scientific papers. This illustrates something that's really neglected in the UK, that recreational angling can actually be a force for good and for conservation. After all, high shark numbers are in our interest. There can be significant mistrust between anglers and scientists, and hopefully this work, made much easier because I have a foot in both camps, might illustrate the advantages of cooperation. Right, this is a bit from tagging data. Blue sharks migrate vast distances annually, so the fish we see off the Cornish coast could be off the west coast of Africa by early winter. They broadly seem to follow the major ocean currents, and though migration patterns differ by sex and size. Here is some real data on the right-hand side here from a program that the SACGB took part with using spaghetti tags. The red indicates female and black male sharks. In total, 1,440 tags were applied between 1998 and 2011, and there were 99 returns, a recapture rate of 6.8%. The average time at liberty was 426 days, and though one female fish that was a mature size when tagged was recaptured 18 years later, indicating that the fish was probably over 23 years of age when finally recaptured. On average, the fish were recaptured over 3,000 kilometres away from first point of capture, with the nearest caught locally by another loose shark boat, and the furthest was actually off the south coast of Venezuela, over 7,800 kilometres away from capture. Given a straight line speed, which obviously isn't true, the speed of these fish varied between 0.3 to 45 kilometres per day, with one fish being recaptured off the coast of Maine 80 days later. This is the boring stuff. Overall, the fishery was highly dominated by female fish, with over 94%. Only 8.61% of females and 6.92% of males were considered mature when captured. It's also possible to generate some useful growth data, much of which I won't bore you with again, but on the right-hand side of the VB growth parameters and confidence intervals, thanks to Elephant and my old friend the jackknife, and these were broadly in line with other studies. Right, these are the catch per unit effort of blue sharks from the Shark Angling Club of Great Britain data from 1953 to 2009. You can once see that the initial high catches decreased in the early 1960s, and despite the occasional better year, never really increased to these levels until after 2010. The question would be, does that actually represent where the stock is? Well, the answer is probably not quite. As you can see, the environment actually influences the catches. The red bar there, the blue bar there is actually the cold winter of 1963, and the red bar indicates one of the warmest summers. So the environmental changes can actually affect the number of fish we capture in the southwest of England. The Vass paper in 1990 showed that shark captures in the UK were highly temperature dependent, especially the temperatures in the spring months. And Mitchell et al. Also showed that the levels of algae in the water and the presence of mature tidal fronts influenced the local distribution of sharks. But my question was, what about bigger environmental changes that affect the oceans? In areas miles and miles away, do they actually change the number of blue sharks we see in the southwest of England? Surprisingly, on a yearly basis, sea surface temperatures anomalies had little influence on between yearly catches, and though these do affect the between monthly catches. The yearly chlorophyll averages for the summer months had little influence. These factors seem to control mainly local distribution rather than migratory patterns. I've just overlaid some salinity data from the North Atlantic taken from the Holiday et al. Paper. They show that they showed freshening events in the North Atlantic and managed to reconstruct a time series going back to the 1950s. And there seemed to be a plausible relationship between the low salinity events in the North Atlantic and the explosion in blue shark numbers seen in the mid-2010s. So, why do changes in the oceans thousands of miles away affect the blue shark numbers we see in the summer in the UK? The most likely explanation to me would seem to lie with the current systems in the North Atlantic and changes to the AMOP. I'm going to skip the bit explaining the thermohaline circulation, as most of you probably understand it better than I do. This slide is it's adapted from Holiday et al. And illustrates how changes in salinity affect the strength and position of the currents. In the graph, the differences in the amount of wind stress between the Arctic Atlantic and more lower latitudes leads to a negative salinity anomaly off the southeast coast of Canada via the cold Labrador current, which in turn strengthens the northern arm of the North Atlantic current. This seems to result in less sharks seen in the UK waters. When we see an increase in blue shark numbers in the UK, increased rainfall and ocean heat loss in the northern waters lead to high salinity off Canada and low salinity events in the North Atlantic, which result in a slowing of the northern arm of the North Atlantic current and a strengthening of the southern arm, which is closer to the UK. Right, so if numbers of sharks that reach our waters may not actually be directed to stock levels, how can we compensate? We model them. So initially I used boosted regression trees to identify variables of importance, and then use generalized additive modeling to try and take out each effect and just leave the differences between years, which then should better represent the change in stock. In the model, the salinity changes in the North Atlantic explained an astonishing 24.52% in the variation in blue shark numbers, whilst the Atlantic metadecadal variation and the North Atlantic oscillation explained about 4.5% each of these differences. However, when you closely look at the data, you can see that the blue shark catch per unit effort lags the AMV by up to 10 years. And there is also some covariation between variables. This pattern was increases in shark numbers was also, as the bottom graph showed, seen in other ports in the southwest of England, and it was also seen in the fish stocks in the Celtic Deeps. So, what if this this model of yearly captures looks like when I actually remove the factors? The red lines on the left-hand graph indicate significant reduction in shark catches between years, and the blue lines significant increases. I use the finite differences method to do this, so if anyone gives a damn. The data suggests that there's been a real increase since the mid-2000s, but as with all of this stuff, this would need to be confirmed with other data sources before concrete conclusions can be made, and this data has been and will be submitted to ICAT. So, has the global blue shark stock recovered? Well, maybe. What we can say with some certainty is that the number of fish reaching the UK in recent summers has increased. And the data does look similar to that collected by the Irish Recreational Tagging Programme. Why might a recovery occur? Well, being hopeful, has the main pressure, which is the longline fishery, decreased, and have the limits on finning actually had an effect? That is an interesting question, which may or may not be true. Has predation increased? An example of this would be that the stocks of one of the blue shark's major predators, the mako, are in a perilous position, and the NOAA data suggests that indeed makos were in the blue shark pupping areas at the same time. Have increases or changes in prey distribution had an effect? Well, blue sharks and mackerel numbers correlate well, but strangely, only for mackerel caught in the summer between Norway and Iceland, which actually does suggest a common cause. However, both anchovy and pilchards have returned to the southwest in large numbers, but unfortunately accurate data isn't available for this ICES area. The recent surge in blue shark numbers in the UK has coincided with, as Tom will talk about later, the return of bluefin tuna, and even the elusive thresher shark, as Doug said earlier. There is even some indication of a recovery in pool beagle numbers recently, but this is probably due to the commercial zero TAC for the species. However, as we all know, ecology is a slippery beast, and rarely does one factor explain the fate of a species, especially one that travels as widely as the blue shark. Here is just some details of the papers, either been submitted or are in submission, and some contact details there. So, that's the end of it with my Welsh Rugby Union rugby ball, and questions, please.
Okay, thanks, Simon. Just ask you, yeah, there we go. If you'd like to share your camera, there we go. Okay, so yeah, we haven't got a huge amount of time for questions. We had a couple that have come up in the Q&A box. So firstly, Bethan McGuinness-Deans asked, why do you think there's such a huge difference between the numbers of male and female sharks being sighted?
They have been known to, they have always segregated by sex. Typically, the male sharks are the other side of the Atlantic, especially the mature ones. But interestingly, in the last 5 years, we've seen a massive increase in big males in the UK.
Okay, well, thanks for that. Borbell has said, is there still a NOAA shark tagging management program?
NOAA still operate a program. Many skippers take part of it, and though the legality of it in the UK is probably dubious, but it happens. Okay.
He's also asked, are there significant interactions between fisheries and blue sharks in the Atlantic?
Certainly, yeah. I mean, the long lining fleet claimed that blue sharks were bycatch, but this has recently been changed, apparently. the tagging data that I shared probably reflects where people are fishing for blue sharks either deliberately or the interaction between the blue sharks and the swordfish fishery.
Okay. And last of all then, so Bob's also asked, where are the pupping areas off Portugal?
They're considered to be between the Azores and the Grand Banks, and though in recent years, especially last year, there were a reasonable number of pups, newborns seen in the UK.
Okay, brilliant. Okay,
if I can come in on that, about 10 years ago, there was a pup caught in the Scillies.
Yeah, I had probably 15 reports of newborns this year.
Wow. There you go. Okay, so that's obviously some significant changes going on. And so thank you very much for that, Simon. There aren't any more questions in the Q&A, but obviously if people do have questions, then you can ask them throughout, and we'll try and hopefully, Simon, you'll be able to pick some of those up in the Q&A.
Thanks very much.
Okay, thanks very much. Okay, so up next we have Ben. Are you there, Ben?
Hello, can you hear me okay?
Yep, I can hear you. So would you like to share your screen? That works.
So you should be sharing now.
Yeah, that looks good.
Okay.
Okay, I'll leave it to you then.
Thank you, Jack, and thanks to Doug for inviting me and for the Southwest Marine Ecosystems for hosting this session. So I'd like to talk about some work that's really ongoing in the southwest of England, and I've added to the title via Scotland because most of the work on this topic I've done in the past has actually been up on the west coast of Scotland, and I'm sort of transferring some of that work down here at the moment. So much of what I'll be talking about will be previous work in Scotland that's being applied to this area. So everyone in the southwest is going to be familiar with this massive annual migration of Sarcopterygians to the shore. So people flock in great numbers, particularly to sandy areas, and seek out the shallow, warm waters in the spring and summer. But of course, there's a parallel migration that's happening beneath the waves. So young fish, immature, juvenile fish move inshore and use shallow inshore areas during their first years of life, and particularly during their first summer. So the adult fish are often what we see and we like to catch and we eat, but every adult is dependent on a precarious process of development through production of huge numbers of eggs, where there's very then high mortality during early life. And it's this mortality during early life which regulates the size of the populations. So these early life stages that come and use these shallow areas are really important for the population dynamics. So the juvenile stage, in many cases, is where key population processes interact with our human uses of the coastal zone. So if we can understand what makes a good habitat, we can ensure that these important habitat functions are protected to ensure that we have viable and sustainable fish populations. And in many cases, we don't know all that much about the juvenile life stages of fish, much less than the adults, because they tend not to be found in areas or in habitats that are fished. They're not usually caught in the usual commercial or recreational fishing gear, and they tend to be quite small and cryptic. So areas that are important to juvenile fish are often called nurseries, and we usually think about those iconic vegetated habitats, things like mangroves, seagrass, salt marsh. But actually, many of the commercially important European fish rely on sand or mud, more do so than vegetated habitats, especially in the nursery stages. A good example are the flatfish. So the flatfish are an important component of UK fisheries and include some of the highest value species in the industry. So about 25,000 tonnes of flatfish are landed by UK vessels each year, which amounts to about £80 million. And this is about 8% of total fish and shellfish landings. And 2/3 of the UK flatfish landings are from species that rely exclusively on shallow sandy areas during juvenile stages. And when I say shallow areas, I mean less than meters in depth. So other areas beyond these iconic vegetated habitats can also be important. Things like sandy beaches and sandbanks, but also human areas which have got heavy human impact as well. So habitats play a role in determining the population dynamics of fish. So we need to identify these habitats, if we're going to ensure that they're monitored and protected. Now, for about 20 years in the US, there have been efforts to identify essential fish habitat. So essentially identifying these key habitats that fish require. But one of the challenges is actually defining what we mean by essential. So the yellow area here shows essential fish habitat that's been defined for summer flounder. Basically, the map was built on the presence absence data. So wherever summer flounder were found, that square was filled in as being important for habitat for summer flounder. The issue is, in this example, the entire eastern seaboard is essential. And the trouble is that where everything's essential, nothing becomes essential. So if our criteria for identifying essential fish habitat is too broad, we've got little power to discriminate between habitats and set conservation priorities. So what we need are some robust metrics that can discriminate habitat quality and allow conservation to be prioritized on habitats that need it most. So a number of criteria are sort of suggested, starting with sort of the most basic, it's just presence or absence data. If we can do a bit better, we can get densities, and that tells us not only whether a fish species is present, but how many are present. But of course, just having lots of fish in a habitat doesn't necessarily mean it's a good habitat. If the fish aren't growing or surviving in those habitats, it could be completely the reverse. So we also need to know how well the fish grow, survive, and reproduce in these areas. And obviously for the juvenile fish, the growth and survival is the key thing there. And finally, think about production and essentially piecing together density, growth, and survival to work out how productive these areas are at producing fish. So number 1 on these and 2 on this list here, the presence, absence, and the density, is where most studies of fish habitat and essential fish habitat tend to focus. Getting to level 4 is actually quite challenging. So what much of my work is focused on is on actually looking at this level 3 here and trying to understand not only the density of the fish in a habitat, not only how many there are, but also how well they're growing and surviving. So what do we actually know about growth variation in juvenile fish? And this is a debate which stretches back many decades. What controls the individual growth of juvenile fish? And perhaps none more so than in a particular species of flatfish, the plaice. And then in this species, debate has revolved around the importance of food limitation in controlling the growth of the young fish. So plaice spawn in the winter, and then they lay eggs which are pelagic and develop into pelagic larvae which drift and eventually in the spring arrive in shallow sandy areas such as this beach on the west coast of Scotland. And they'll spend the summer in, as I say, shallow areas, so typically less than 5 meters deep, but most commonly in just sort of knee deep or waist high water essentially. And they'll migrate in and out with the tide, and they'll feed on small invertebrates, the siphons of bivalves, the tentacles of small crustaceans. And so they'll arrive about postage stamp size, and they'll leave when they're about at the end of the summer and about the size of your palm. And they might come back in future years, but it's mainly the first year of life that really relies on these very shallow inshore areas. So observations of abundant prey in these areas led to the suggestion that fish tended to have lots to eat, and growth was only limited by temperature. And this was backed up by a number of studies that found rates of growth matching maximum rates expected from lab studies. Okay, so these red dots show all the studies that found this maximum growth rate. However, at the same time, other studies detected slower growth rates consisting of density dependent or density independent food limitation. So a number of different findings. How can we reconcile these conflicting findings? And I guess this is the slippery beast that Simon was talking about. Well, the issue here is that these are all studies that were undertaken at different times and in different locations. So if the factors controlling growth vary in space and time, it's hardly surprising that we're going to find different results. So what we actually need to do is assess the patterns of growth in much more detail first, in a spatio-temporal context, before trying to understand the causes. So we need to move from sort of a yes, no, is food limiting growth or not, to where and when is limitation occurring and why. And in order to do this, we need sensitive measures to measure growth, methods to measure growth over broad areas. So it's actually quite difficult to measure growth in wild fish. So you can, if you want to measure growth in the lab, it's fairly straightforward. You can take a fish, measure its size, leave it, feed it for a couple of weeks, and then measure its size again. And the change in size will give you the growth rate. But in the field, it's much more difficult. You can get that initial measurement, but then you release the fish and chances are you're not going to see it again. It's off. So you can't ever get that second measurement. Now, fortunately, there are many things that correlate well and relate well to growth rates in fish. And one of those is the content of RNA in the fish tissues, So essentially the biochemical composition of the tissues. So in the lab, if you can measure both the RNA concentration in the tissues and the growth rate, you can develop some sort of relationship between these two things. So you can actually do experiments where you grow fish at different rates by feeding them this of different amounts, and then you measure their growth rates and their RNA content. You do that at different temperatures because this relationship is temperature sensitive. And once you understand this relationship between these two things, you can use that as actually a tool to measure growth in the field. So you can go out, collect fish of an unknown growth rate, measure the content of RNA in their tissues, and then using this relationship, work out what rate they should be growing. So this essentially provides a proxy. These RNA-based indices provide a proxy for growth that we can use to measure, to predict growth rate of fish in the field. Individual growth rate, quite sensitive and high throughput method that can be applied over broad areas. So we applied this on the west coast of Scotland. So here you're looking at 7 different beaches on the west coast of Scotland in 2 different years. So the 2 different rows here, essentially 2005 and 2007. And we're looking at growth rates from the middle of July to the middle of September. So each of those panels, that's the x-axis there. And what we can see here, each point here is an individual fish, and this has been fitted with a sort of best fit model, essentially. And what we can see here is the time in the season, unsurprisingly, makes quite a big difference. So towards the beginning of the season, most of these sites, fish are growing quite rapidly. These juvenile place are growing quite rapidly. But then gradually over the summer, growth rates decline until by September time, the fish really just aren't growing at all. They've essentially stopped growing. And actually the red band here shows the range of expected maximum growth rates. So towards the beginning of the summer, these fish are growing quite rapidly. There are some exceptions, and we're sort of looking into those, that like Caylesport and some other sites that show slightly different patterns. But overall, there's quite a consistent temporal decline that we found in our work on place on the west coast of Scotland, but it's also found in other species and other place studies in other parts of the range using different methods. Okay, so this partly solves the question about what's controlling the growth of these young fish, because actually it depends what point in the season we're talking about. The temporal context is important. During early summer, fish are growing quite rapidly, and yes, they might be experiencing maximum growth rates, but towards the summer, something's limiting their growth, and increasingly they're becoming probably limited by food. But we still don't know why. So we know that we can see this pattern, we can explain these conflicting findings, we don't know why. So we took a wider look at growth rates in August when growth is limited. And And so this map is showing, again, the west coast of Scotland. The size of the points represents the relative growth rate. You can actually see there's substantial variation among sites and among years, and even very adjacent sites can have very markedly different growth rates. So our traditional explanations of this didn't really do a good job at explaining this spatial growth variation. So there was a small relationship between growth rate and the density of the fish in these beaches, but there was no effect of temperature. And actually some of the physical characteristics of beaches were much more important. So there was a strong correlation with wave fetch and with the tidal range of the beach, which we know sort of have an overarching influence of the ecology of sandy shores. So that brings us really to the southwest, and the work we're doing at the moment down here is to try and work out how these physical factors influence specifically the growth rates of young flatfish, but also their habitat quality in general. So we're using basically the southwest as a big natural laboratory, because we've got a range of different beach types down here, from very sheltered, reflective beaches to more exposed, dissipative beaches. And we're looking basically at how these vary in their habitat quality for juvenile flatfish. So I'm not going to go into the results of this work now, but just to say we're finding basically 3 main juvenile flatfish that are using these locations. It's not just all about the place down here as it was up in Scotland. We've also got good numbers of turbot and a smaller number of sole, and also a smattering of other species as well. A PhD student of mine, Anna Pearson, has conducted basically 2 years of sampling at 17 beaches throughout the southwest, and we're trying to correlate patterns of variation in abundance and of growth with the physical environment. And now the next step really is to turn this into a more long-term monitoring program to get an idea of interannual variation in numbers and growth of juvenile flatfish, and that's something I'm continuing with other students. So I'd like to just finish there and thank the people who funded this work and all the people who've helped and collaborated going out collecting juvenile fish at various institutions, and particularly at the University of Plymouth. So thank you very much.
Okay, thank you. Thank you very much, Ben. I think we don't have very much time for questions. We've got a couple of very, very quick ones, if you can kind of take them quickly. Well, I'll say that we'll take the first couple of questions, and then if you're happy, Ben, to kind of
Sure.
check in the Q&A and maybe answer some of those questions offline afterwards, that'd be fantastic.
Yeah.
So firstly, Matt Bell has asked, how do you collect RNA from wild fish, and what are you looking at in the RNA to determine growth rate?
How do you collect the RNA? Is simply we have to take a sample of the fish, and they need to be frozen and stored, and then we take a sample of the muscle tissue, essentially. We're looking at the bulk quantity of the RNA. We're not looking at any specific RNAs. The bulk quantity is quite a good general measure of the machinery for protein synthesis. So it's basically telling us how many ribosomes there are. That's what makes proteins, and ribosomes are made up of RNA. Okay.
And Thomas Stamp has said, has there been many mark recapture studies on juvenile flatfish, and could these also be used to measure growth rates at different beaches?
Thanks, Tom. Yes, there have been some. There's actually been quite a big study of mark recapture of flounder in the Tamar around here, but also a number of studies in other locations. Up on the west coast of Scotland, there's been some work tagging fish, and yeah, they do provide ways to measure growth rate, and it's a good, accurate measurement of growth rate. But in order to, I guess the difficulty there is that there's a lot of investment of time in order to get a small amount of numbers. So if you've just got a 10% recapture rate, for example, you need to tag, hundreds of fish just to get tens of samples. So it's not really something that can be done on mass at a number of different nurseries. It's more something that's been applied at single locations.
Yeah. Okay. There's one more question from Susan Fraser. But what I'll probably do is I'll read it out, but I'll leave it to be answered in the answered section, because it's kind of a philosophical one as well, I think. So, Seaspiracy is a new hard-hitting Netflix film about the commercial fishing industry. Do you think it will be as influential as David Attenborough's Blue Planet films have been? Has anyone seen it, and any comments? So I think that's a kind of a broad one. I don't know if you want to say a couple of words on that, or if you'd like to just take that into the Q&A chat.
Something that I definitely would like to see. It's not something I've seen, but yeah.
No. Okay.
Yeah. So thoughts about that from everybody would be great.
Yeah, brilliant. Okay.
Thanks.
Well, thank you very much, Ben. That was a fantastic talk. We're going to go on quickly over to Tom now. Tom Horton is going to be talking about Atlantic bluefin tuna. So are you there, Tom?
Yeah, just sharing screen quickly. Can you see that?
Yeah, we can see that. That's great. I'm going to pause recording now, but that was fantastic. It was a great overview. Obviously, we're running very much behind time now. There's a couple of quick questions, which I think would be, two of them I think are quite similar. So the first one, Michael Poulston and Mark Darlison have both asked questions which are quite similar about proposals to open up big game fisheries for bluefin tuna. So Michael's asked particularly, do you think this should be encouraged in the southwest? And Mark has asked, does it stress or damage the fish badly? So what are your thoughts on that?
So I suppose the first question about whether big game fisheries should be kind of encouraged or not, well, we have quite a clear goal in terms of our work, which is ecological work. So we don't really kind of tie into the kind of whether it should or shouldn't happen. And those questions should really get directed to DEFRA or one of the organisations that's managing that, and perhaps Simon might be able to feed into that one. In terms of this kind of ongoing question of whether capture damages fish, I think that we could probably all safely agree that fish that's caught by an angler is never going to be better off after the event, but that there's definitely ways that anglers can be more sympathetic to improve post-release survivorship and sort of minimise the effect of capture. And again, Simon's alluded to that, and it's an ongoing topic. And I'm sort of pleased to say that it's a real it's at the forefront now of this kind of like angler fish interaction. So yeah, there's definitely ways of mitigating that.
Okay, thanks. That was a good answer. So Tom Stamps asked, and actually this is kind of related to Bob's question as well, so they're linked together. So Tom's asked, do you know why the tuna numbers have increased in the UK English Channel? And Bob's asked why the recent upsurge in sightings in the southwest. So they're kind of related. So do you think there has been an increase? And Bob's, and connect that, I guess, to Bob's question about why the upsurge in sightings.
Yeah, it's do you know what? It's one of those things that we do tackle this in the paper that's about to come out. Although one of the things that's a real challenge in trying to understand this is the fact that tuna have arrived very recently, and as Simon demonstrated so eloquently with his cracking data set, is that you can start to pull out trends based on climate and stuff like that over long time periods. We've only really got a sightings database of 5 years or so, because there was never a capture fishery for them in the southwest. That said, there are several elements at play here. I think you can't ignore that bluefin tuna have been a kind of management conservation success story over the past decade or so, and the population seems to be doing much better now than it was, say, for instance, 20 years ago. And that is coupled with potentially favourable environmental conditions. So there has been work that, again, ties in, sort of dovetails into what Simon's saying about these kind of large-scale hydroclimatic processes, for instance, the AMO, that make areas either not beneficial, but better sort of areas for fish. And we are in a positive sort of habitat phase in the northeast Atlantic for bluefin tuna, if you were to believe there's long-term indices. One thing that we do point out, I suppose, in the paper, is that actually there is a bit of a temporal mismatch between the onset of these kind of favourable habitat phases and when bluefin came back. So the most recent favourable habitat phase in the northeast Atlantic started nearly 20 years ago. And for those of you that are keen anglers or have keen eyes or read the tabloids will note that actually bluefin have only been here for 5 years. So it was good habitat, but there was no bluefin for about 15 years or so. So what I suppose we'd say is, the Facebook status is it's complicated, but it's likely a combination of multiple factors that are kind of acting in concert to bring them back.
Good. Thanks for that. But one very quick, hopefully last question. This might even be a yes or no answer. So Elizabeth West said, "Brilliant talk, thanks. When tuna enter very shallow water, are they following prey?"
I mean, I guess they would be, yeah. I suppose that, and there's some fabulous videos, in fact, that I did dare show you because they didn't work. But there's a great video of a tuna presumably chasing bass in like somewhere in St Ives Bay, Carbis Bay, and there are some other videos of tuna in harbour and stuff like that. But you imagine that they're feeding. I suppose the one thing I would say is that if you are trying to oversimplify the life of a tuna, they're either feeding or they're reproducing. So once they're outside of those known spawning grounds, say for instance the Mediterranean or the Gulf of Mexico, and there's a few other ones, they are dead set on putting weight on and feeding. So pretty much their entire existence whilst they're here is revolving around feeding. Brilliant.
Great answer. Okay, well, thank you very much for that, Tom. I think we're going to hand over now to Doug for a quick couple of words to finish off. What I will say as well, though, is I'm going to leave the Q&A open now. So if anyone else has got any other questions or any panelists want to stick around and answer any of those, I'll leave those open. I'll leave the chat open as well. So if people want to add their records, then please do. So yeah, thank you very much, Tom. Doug, are you there? Thank you.
Right. Just to say, let's see if my video come on. No, it won't. Okay. Just to say great thanks indeed to Simon, Ben and Tom for 3 most varied and interesting results. Really fascinating to see. But also, altogether, all the material we presented today showed what an amazing ichthyofauna we've got in the southwest now. We've got the largest and the smallest Northeast Atlantic fish, some of the rarest, and yet on the other hand, some populations appear to be increasing, the bluefin tuna, the at larger end, and the variable blenny at the smaller, and also expanding ranges. There's so much there of interest. We need to record it. So please, please, please do send in your observations to Bob or to myself, so we can actually get all this material together and produce the most outstanding Southwest Marine Ecosystems report, annual report for 2020. So thank you very much. Please send in those records, particularly turtles. Bye.
Okay, thank you very much. Thanks for that, Doug. And so, yeah, thank you very much. As I mentioned at the start of the day, we're putting together all of the information for the 2020 Southwest Marine Ecosystem State of the Southwest Seas report. So please share any of your 2020 marine observation highlights of your own. Now the session is ending, you can do that by following the instructions which are shared on the weekly webinar updates emails that Bob sent round. I'm actually going to leave the chat and Q&A open until 1:15. So if you've got anything else you'd like to type in there, then feel free. The next Southwest Marine Ecosystem session is on oceanography and plankton, and the session's taking place on Monday, so Monday the 29th of May at 2:30 UK time. So tune in for that. Finally, I'd just like to say thank you to all the speakers. Thank you, Doug, for opening the session and closing. And thanks also to the Southwest Marine Ecosystem Organising Committee, and obviously the Marine Biological Association for hosting today's session. Most of all, thank you all for joining us, and particularly everyone who shared your insights and your questions during today's session. And thank you very much, and goodbye.